All right, we're going to go ahead and get started. Hello and welcome everyone. Thank you for registering for the End This Forum seminar series hosted by the Cambridge University Biological Anthropology Society, an independent student-led society dedicated to origins research. We offer a warm welcome to our guest speaker today, Professor Terence Capellini. Professor Capellini serves as Professor of Human Evolutionary Biology in the Department of Human Evolutionary Biology at Harvard University. He also leads the Developmental and Evolutionary Genetics Lab. Professor Capellini and his lab are currently focusing on the discovery of genetic alterations that determine the unique human postcranial skeleton and the elucidation of the genetic and developmental architecture underlying the diversity of joint types in primates. I'll now hand the time over to Professor Capellini, and we kindly ask that all questions or comments be saved for the Q&A session at the end. Thanks for joining us.
Excellent, thank you. I'll start sharing my screen. Okay, excellent. And I just want to make sure I have a pointer. All right, thank you so much for inviting me to give this talk. I'm super excited to be here virtually. So let's just get to it. So my lab's focus is really on understanding how the human skeleton kind of evolved from a developmental point of view and what impact the evolutionary history of the human skeletal development has on our disease risks. So we have lots of projects that span many different parts of the skeleton and the body itself. So for instance, we're studying the effects of Neanderthal and digressed regions on immune systems and brain biology. We're studying how humans develop unique thumbs and big toes. We're studying how the knee forms and why that's linked to osteoarthritis risk. We study the genetics of height variation across human populations and long bone lengths across primates. And we focus a lot on the girdles, the scapula and the pelvis. And today what I'm going to do is tell you a story about the pelvic girdle and how we can use our tools to kind of explore the pelvic girdle evolution and development. And speaking of tools, we use a lot of different tools, all kind of geared towards connecting the genotype to the phenotype. If you align humans to chimpanzees, you know there's lots of genetic changes, millions of genetic changes, but which ones are the actual functionally relevant ones that control aspects of human biology and human specific traits is really unknown. So we can use tools from all the different areas of science that you see here listed to really help us develop models on how a particular gene or set of genetic sequences are used to orchestrate the development of a particular novel adaptation in humans. So today, I'm going to use these tools and go through these tools and discuss how we've evolved our pelvic shape from a developmental point of view and how it's linked to disease risk. So let's just start with human evolutionary biology, an area that we're all very familiar with. Now, if we look at the human fossil record, and we compare it to chimpanzees and living humans, we know that pretty early on in human evolution, our hind limbs evolved for walking on two legs. And our pelvis, our pelvic girdle, really shows some of the earliest adaptations to bipedalism. So for instance, you can see Ramidus and Australopithecus afarensis, Africanus, Homo erectus, and Homo sapiens. You can see how we've gone from having iliac blades that are more dorsally, coronally oriented in chimpanzees, to having these iliac blades that kind of rotate and reside in the kind of the parasagittal orientation. And obviously that's important for the function of key muscle groups that allow for walking on two legs. So in fact, over the course of evolution from a last common ancestor, we rapidly evolved our pelvic girdles. And so modern human pelvises, for instance, have these very curved, wide iliac blades that are kind of organized in the parasagittal orientation. So the lateral stabilizer muscles are on the sides of the body. We've extended our pubic rami. We've changed our ischial regions. We've kind of dorsalized them and posteriorized them. And we've made larger hip sockets or acetabula. And these changes occurred pretty rapidly early on in the human evolutionary record. So we also know that if you look at a newborn, these types of changes are already pretty evident in the newborn skeleton, particularly in the pelvis. So the question really is, how has evolution targeted pelvic development? Because somewhere along the line, the pelvis had to evolve these structures, these changes, and it must be through developmental mechanisms. So evolutionary biology tells us like kind of the functional kind of time frame of human evolution and when things happen. But developmental biology of the tool can tell us when in development these changes are actually manifesting. And so let's bring in developmental biology here. So what I want you to understand is that while much research has been done on the mouse model, the mouse is a quadrupedal animal. It walks on all fours. It's small-bodied. The loading patterns are quite different. And so to really understand how humans evolved and humans develop pelvic girdles that are specific and hind limbs that are specific to human bipedalism, we really need to study human embryogenesis. So here you're seeing a developmental series of human embryos up through 8 weeks gestation. We actually study in the lab human embryonic samples that we get through very ethical means from the Robot Epigenomics project. I'll be able to answer questions about this after the talk. But we get human embryonic samples ranging from around 44 days gestation, shown here, to around 56 days gestation, shown here. So, about 2 weeks of gestational period where you see these massive changes in morphogenesis occurring. And what we can do is we can identify during these developmental stages how the skeleton is forming. And so you're probably all familiar with endochondral ossification, the idea that you have a cartilage model that's laid out that then becomes ossified and eventually hardened into bone. And so during this window of development, we see this endochondral ossification program being laid out. And so one of the first things that we wanted to do was actually just look at what a human skeleton looks like at a very early gestational time point. So we used a technique called Alcian blue and alizarin red staining. So Alcian blue, shown here, is staining the cartilage. It stains glycosaminoglycans, proteins in chondrocytes. And the red here is staining calcium in bone, so the hard bone. And what I want you to appreciate is by 8 weeks gestation, the human pelvis already looks like a human pelvis. The iliac blades are already curved in the parasagittal orientation. They're short. The pubic rami are extended. The ischial regions are posteriorized. And while it's not easy to show here, our acetabular sockets or hip sockets are already quite wide. And so we actually have developmentally by 8 weeks gestation, this human form occurring, but at the level of cartilage that precedes bone. That's an important point. It's in the level of cartilage. We actually, working with wonderful colleagues at the University of Amsterdam, we isolated key windows of development of the human pelvis. And what you're seeing here are cartilage reconstructions, not bone, cartilage reconstructions of a developing pelvis. So this is all preceding pelvic ossification. So here, for instance, at early development, 44 days gestation, you see the development of the pelvic girdle. Here's the femur. At one point, the ilium becomes a rod-like structure. Pubis and ischium are beginning to form. And then the ilium takes off in terms of its growth and development. It actually starts expanding in this plane, in kind of the paracoronal plane. And eventually it starts expanding, getting larger and larger, and it starts to rotate in the parasagittal orientation, such that by 8 weeks gestation, you see this pelvis that's kind of beautifully rotated. And just give you an appreciation of what this looks like from above, if we look at a CS20 stage, this is around a 48, 50 stage day embryo, you can see that the iliac blades are kind of paracoronally oriented. But by 8 weeks gestation, just 8 days later, they rotate in the parasagittal orientation. So now they're taking the characteristic human form. And we're actually currently exploring why these rotations happen from a developmental genetics perspective, which I won't talk about right now. So you can see this is just an angulation difference here in terms of this parasagittal orientation. Now, during this window of development, so during particularly this window here from CS21 to CS23 or 50 days to 58 days, what you can begin to see is that you have cartilage development. This is all cartilage. There's no bone here. But about 10 days later, by 67 days gestation, you get the formation of a primary ossification center. This will eventually, the whole pelvis will eventually be replaced by bone. But the ossification center sits dorsally. It sits near where this region is going to articulate with the sacrum, and it doesn't really reside ventrally. In fact, we've looked at very late stage specimens about, let's say, up to about 20 weeks gestation, and we're still seeing evidence that this region here is cartilaginous. It's made up of chondrocytes, but not bone. So in other words, there's a stalling of the ossification front and this region does not get ossified right away. And so we call this region right here the anterior zone of chondrogenic growth. We also refer to it as the chondrogenic zone of protection. It's just a zone that maintains its cartilaginous state, and therefore the cells can grow and proliferate. If we compare this to the mouse model, they have ossification that goes right across the ilium from the beginning of development. Right when you start seeing ossification, it is occurring across the entire iliac blade, unlike what you see in the human form. So developmental biology really allows us to identify that natural selection has targeted early human chondrogenesis, cartilage development, to accommodate the adaptations that we see later in life. So developmental biology tells us the tissue context and the timing, but now we want to know more about how this process is orchestrated by genes and regulatory regions in the genome, and whether or not there are important places in the genome that humans have evolved to shape these developmental architecture of the pelvis. So we use tools from functional genomics, which are tools that allow us to look at genes and regulatory elements in the genome. So working with my wonderful graduate student, Marielle Young, who graduated last year, we worked together to extract cartilaginous regions from a developing human embryo. And so we can take the cartilaginous region, we can take the ends that are cartilaginous, and for each chondrocyte cell that sits within these ends, the chromatin that actually has genes and regulatory regions can be assayed. So you all know that the regulatory regions and genes orchestrate developmental processes in cells. And so we can use a technique called RNA sequencing on chondrocytes extracted from these regions to find genes that are expressed in these cells. And we can also use another method called ATAC-seq that allows us to look for regulatory regions, enhancers, switches that turn genes on and off in particular cell types. So let's start with the RNA-seq approach. So we've acquired a lot of human embryonic samples, and we do the same thing on stage mass mouse embryonic samples. We do this on not just the skeleton, but other organs like the brain. And for every one of these lines is pointing to a different anatomical structure that we assayed using RNA sequencing and ATAC sequencing for genes and regulatory regions across human development and across mouse development. So we've done a lot of different tissues, as you can see down below in the totals, and we have a lot of different, we have a lot of power to examine regulatory sequences and genes in the genome. So regarding RNA sequencing, which again is a method to identify genes that are expressed in tissues, we can take the human pelvis during development and extract chondrocytes from the ilium, from the pubis, from the ischium, and even from the hip socket. And we can extract RNA in these separate pools and then do a large number of them and identify regions of the genome that are expressed. So this is a PCA, principal component analysis of the genes that we're getting from different tissues. And what I want you to appreciate is that on PC1, about 21% of the variance is described by where in the pelvis the genes are coming from, or genes are expressed. So for instance, here in blue are all the ilium cell samples that we used. Here in red are acetabulum and pubis. The ilium actually forms part of the acetabulum. And on the left side here are both the ischium and ilium. So what we're seeing are changes in gene expression based on where superior versus inferiorly you are in the pelvis. But what we also noticed is that if we compare female samples to male samples, we already see early on differences in gene expression between male and female samples. Now one would like to believe that this could be related to sexual dimorphism in the pelvis and the skeleton in general, but I want to kind of emphasize that if you took any human cell from the body and looked at gene expression differences between sexes, you would actually see this. You would see these differences already. So it might not be specific to say sexual dimorphism. The other thing you all know in this audience is likely that sexual dimorphism occurs much later in development. It doesn't mean this is not important, but for this talk, what we do is correct for differences in sex of the individual samples and moving forward. So what we can do with the RNA sequencing data is actually identify genes that are either upregulated or downregulated in, say, the ilium here by this green bar, compared to all of the other parts of the pelvis. So this gives us genes that are kind of uniquely turned on and off in the in the ilium. And you can do this for each of the parts of the pelvis, right? I'm just showing you the ilium results. And so here, for instance, in this plot are gene names on the x-axis. And this is like how well expressed they are across the different parts of the pelvis. And here you see in red expression in the samples of this gene, for instance, in the ilium compared to all the other parts of the pelvis. And this is kind of cool because you can identify genes, for instance, this gene EMX2, which is a major transcription factor that in mouse, when you knock out this gene, it ends up, you lose the ilium, but the rest of the pelvis is normal. So we see the same patterns in humans where, for instance, the ilium expressed lots of EMX2, but the rest of the pelvis does not. So this approach of RNA sequencing really allows us to find genes that are important, likely important to different parts of the pelvis. That's the take home of this. Now, we've also done an analysis of changes in gene expression that occur in each part of the pelvis across developmental time. Now, remember, I talked earlier about how during development of the pelvis, the ilium particularly, there's a zone of chondrogenic protection. It's cartilage cells that are here that do not become ossified. So we can actually look and take cartilage cells from here and cartilage cells from here, do RNA sequencing, and look for genes that are changing, either going up or down in expression over this developmental window. And when we do this, we found 261 differentially expressed genes that are changing over this time course. What is striking is that the genes that are being downregulated or turned off during this window as you move through time are genes that are involved in ossification. So this is kind of commensurate with our observations that we don't see ossification occurring in these regions. It's being shut down. On the other hand, genes that are already turned on, but are turning on even higher, are genes involved in cartilage development. So this zone of chondrogenic protection or anterior zone of cartilage growth is really a zone that's downregulating ossification genes and upregulating cartilage genes. And that's really important. So the RNA-seq analysis that we've done on the developing ilium across time really matches the histological and the anatomical changes we're seeing over development with the stalling of the ossification growth front. Now we decided to do other types of assays that allow us to get at this kind of question of anterior growth zone a little bit more. So we've actually acquired and started using a tool called multiomics and spatial transcriptomics. And I won't go into the details here. What spatial transcriptomics particularly does is allows you to look at histological sections of a sample and identify all the genes expressed in the cells on that histological sample. So it's like a cool two-dimensional map of gene expression. So we did spatial transcriptomics across developing human pelvis, and I'm only going to show you some sections from this stage. And this stage is important because this is the stage where you have this anterior growth zone occurring, where ossification is really starting in the back and anterior growth zone is occurring in the front. So we can actually start looking at histological sections. So this is a histological section. Here's the human pelvis, right? Here's the embryonic stage that we're looking at. And this line here depicts the embryonic section that you're looking at. So you're looking at a coronal section. And here's this zone, right? And what we can do is look at all the genes that are expressed across this entire tissue, all these tissues, and then based on the gene expression patterns, identify what cell types they are. So it's kind of like blind to the cell type until you look at all the genes, and then you know that the genes correspond to those that are expressed, for instance, in blue chondrocytes or in muscle cells, right? So this is kind of a cool approach to look at gene expression three-dimensionally. And this is looking at all the genes that are kind of expressed across the cells in each region. So chondrocyte zone here, and these are all muscles that are anchoring down to the developing pelvis. And if we go deeper into the pelvis, so now we're going deeper in, here's that zone of chondrogenic protection still, and then there's more muscles that are forming. If we move in further, you can start seeing here's the pelvis, here's the ilium. This is part that we're looking at. This is now developing a part of the hip socket right here. And you can start seeing that we're not just seeing cartilage cells, but now we see some pre-osteoblast cells, cells that will become eventually osteoblasts, but they're not yet. And we go deeper in here. Here's deeper into the pelvis. You can still see the posterior part of the iliac blade, this part right here. But then you can also see the ossification fronts and the developing pelvic hip joint. You also start seeing parts of the pubis and ischium. And we can go even further and you can see this more morphology. So spatial transcriptomics allows us histologically to look at gene expression across cell types. And I just want to kind of point out that if we look at markers, genes that are kind of characteristic of a particular cell type, what we can see early on in that early chondrogenic zone of protection, that zone that's like maintaining itself as cartilage cells, is that it expresses this collagen chondrocyte marker, collagen 9A2. It's a marker of chondrocytes. And as we move through, that marker becomes downregulated until you get to the tips of the other parts of the pelvis, like the pubis and the ischium. If we look at other markers of cartilage development, collagen 2A, we don't see it in the zone here. We see it in this region here. This is telling us that this particular region is still being an active cartilage zone, whereas hypertrophic cells are not actively dividing. They're actually becoming large. They're exiting the cell cycles. And then we can move further in and we can see that we start seeing osteoblast markers and downregulations of some of those osteoblast markers in the cartilage model. So I guess what I'm trying to say here is that the spatial transcriptomics analysis really allows us to see histologically that growth zone, but in terms of the anterior growth zone, in terms of all the genes that are expressed. So with that in mind, I've kind of outlined that we can identify genes that are expressed in different parts of the pelvis. I can show you that there's this anterior growth zone that expresses a unique set of pelvic chondrocyte genes that are being maintained, and they're allowing for growth. What we really want to know is, what are the sequences that turn those genes on and off? And you can kind of think about this as like a light bulb analogy. The gene is like your light bulb, and the light switch on the in your room is like the regulatory enhancer, or the switch that turns that light bulb. So you might go to the store and buy like 20 GE light bulbs that all 60 watt, and you might put them in individual rooms of the house, but they're all the same light bulb, but you have different switches. You have a bedroom switch and a bathroom switch and a kitchen switch. And when you turn on that switch in the kitchen, it doesn't affect the switch. It doesn't affect light in your bathroom. So switches can have anatomical specificity. And so we can use an assay called ATAC-seq, which is a method that allows us to look for regulatory regions in the genome. And we can do this on different parts of the pelvis. So for instance, we can take pelvic chondrocytes from the ilium, the pubis, ischium, and acetabulum. We can separate them and identify regulatory elements unique to these individual regions or shared. And we can also do this on other tissues as well to see how specific those regulatory elements are. And so, again, I just kind of reiterate what's important about these switches. And let me just give you an analogy. So on the right here is a mouse embryo. And you can see that a gene here labeled in dark blue is expressed in the head, in the limbs. These are limb buds. This is the forelimb and the hind limb bud, and also in the axial region. This gene, if you mutate this gene, the coding region of this gene, it would affect this gene's function in the head, in the limbs, in the tail. That's actually pretty bad. You don't want to do that. There's a lot of pleiotropy. But we know that there's individual regulatory switches that turn this gene on and off in the head, in the limbs, and in the tail. So modifications to an individual switch can turn this gene on and off, or regulate its level only in a particular tissue without affecting the function of that gene in other tissues. So different switches allow for independent control of different body parts, and these regulatory switches allow unique body parts to change evolutionarily without affecting another body part. So finding switches is important from an evolutionary lens. So we've done our regulatory analysis, identifying regulatory regions in the ilium, in the acetabulum, in the pubis, in the ischium. And if we put them on this plot here, what you can see is that majority of regulatory regions are actually shared across different parts of the pelvis. So here, for instance, 9,658 regulatory regions are actually found. They're active in ischium, acetabulum, ilium, and pubic chondrocytes. So they're not specific to an individual part, they're kind of shared. And that kind of makes sense because a chondrocyte is still the same type of cell if you're in the ischium or the pubis. So there's a lot of regulation that's going to be conserved. In the same way, there are a lot of genes that are likely shared in their expression patterns. But we can see, for instance, for ilium, that there's 2,800 regulatory switches that are only found in the ilium and nowhere else. And likewise, there's 4,000 of them that are found in the pubis. There's about 800 in the acetabulum and 700 in the ischium. And these are anatomically specific regulatory regions not found anywhere else in the skeleton. So this approach of functional genomics allows us to find genes that are expressed in individual tissues, and ATAC-seq allows us to find anatomically specific regulatory switches. So with that in mind, we've now gone through the evolutionary context, the developmental context, and the regulatory and gene expression context for the phenotypes we're interested in. Let's think about how these regulatory sequences might change across primates. So we can use comparative genomics, the next tool. So the question we have is, how has evolutionary selection, natural selection, targeted regulatory regions that shape the pelvis to allow us to be bipedal, to allow us to have evolved bipedality? And so the lowest hanging fruit in this analysis is really identifying regions of the human genome that acquired more changes at a particular sequence than the same sequence in chimpanzees, gorillas, and orangutans. So that means that there's regions where there's more nucleotide substitutions along the human branch compared to the same sequence in chimpanzees, gorillas, and orangutans. And these are called human accelerated regions. They're accelerated because there's more changes in the human lineage than other primates. And there have been different methods to identify these human accelerated regions. And there's about 3,000 of them across the genome. So we can ask, do any of these 3,000 regulatory regions, accelerated regions, interact or intersect with our bone switches? Like, so for instance, our ilium regulatory regions that are unique to the ilium, or our pubis regulatory regions. And if a human accelerated region, or HAR, overlaps with one of these regulatory regions, that suggests that the regulatory region was kind of like acted upon by natural selection. Now, just to put this in perspective, human accelerated regions are found not just in me, they're fixed generally in everybody on this call. Everybody has the same sequence at a particular position in the genome, and even Denisovans and Neanderthals generally have the same accelerated sequences that we have at these HAR positions. So they're very ancient events, so they should reflect past positive selection. So we're going to ask the question whether or not regulatory regions that are specific to individual parts of the pelvis, and here I'm going to bring in the knee, are enriched or have more HARs than expected by chance by comparing any genomic sequence in the genome. So earlier, people have discovered that regulatory regions from the human brain are highly enriched for overlap with these HAR regions. There's more HARs in brain regulatory regions than expected by chance. So we performed ATAC sequencing regulatory region detection in the brain and found the same thing. Other people have discovered that blood cells are not really enriched for human accelerated sequences or HARs, and we found the same thing in our analyses. Let's look at the human knee. In 2020, we had a paper in Cell where we identified that regulatory regions unique to the distal femur, they're found nowhere else in the skeleton, okay, are highly enriched in human accelerated sequences, as are regulatory regions unique to the proximal tibia. So above and below the knee, we have regulatory regions unique to those tissues that are enriched for HARs. Now, if you ask, what about regulatory regions that are shared across the knee, that are used by chondrocytes in the distal femur and chondrocytes in the proximal tibia? And these knee common regulatory regions are actually depleted. They're below the line. They're depleted of HAR overlap. So they're not going to be under positive selection. Now, what about the human pelvis? This is really interesting. What we found is that regulatory regions shared across the developing pelvis. So a regulatory region that might turn a gene on in the ilium, ischium, pubis, and acetabulum, or the part of the pelvic common regulatory architecture, they're highly enriched for human accelerated regions. This suggests to us that the three-dimensional shape of the pelvis is obviously under selection, but that we're seeing it on the genome-wide regulation of shared regulation of different parts of the pelvis. We also see, though, that regulatory regions unique to the ilium, pubis, ischium, and acetabulum, these are switches that are not found anywhere else in the skeleton. They're only unique to the ilium or and vice, across the other tissues. These are also enriched for human accelerated regions. So to summarize this, we see evidence in the knee and pelvis that there's past positive selection that operating on the genome-wide regulation of these structures. And we see particularly within the pelvis that the regulatory regions shared across parts are under positive selection, thereby suggesting some three-dimensional anatomical process. If we take the human accelerated regions, the regions that are kind of overlapping regulatory regions for the ileum, we found 107. There are 107 positions in the genome that are regulatory regions that overlap with human accelerated sequences. And if we ask what biological terms are associated with the genes that they're nearby, we get the term endochondral ossification. So this matches very nicely with what we found anatomically, that there's a delay in chondrogenesis in the ileum and ossification in the ileum, that there's actually this growth zone in this particular area. If we look at the human accelerated regions overlapping the acetabulum or the ischium or the pubis, we're not seeing an enrichment of any specific functionality. They're probably just doing different things, but not working together as a functional complex. So the next thing we wanted to do was we considered HARs. These are these low-hanging fruits, these regions that are really evolving rapidly along the human lineage. But we want to not constrain ourselves to just HARs. So we developed kind of a prioritization method to identify regulatory elements or enhancers that are nearby genes that show much change. So this method works in this way. For every gene that's, say, differentially expressed in the ileum compared to all the other tissues, we are looking at the regulatory elements that are nearby, about within 100,000 base pairs. And we can, for each regulatory element, we can determine how conserved is it across mammals in general. That would tell you if it's highly conserved, it's likely functional across mammals. If it's not, it's likely divergent. We can then look at human versus chimpanzee and gorilla nucleotide substitutions. This is %ID, nucleotide changes. And then we can ask whether or not in the tissue, like the ileum, do we see it acting as a regulatory switch uniquely compared to the other tissues in the body? And using this method and ranking each of these, each enhancer for each of these scores, we get a rank aggregate method that allows us to identify the regulatory sequences that are under particular conditions. And the condition that we were most interested in is identifying the regulatory regions that are near genes that are most divergent, where the regulatory region is most divergent between humans and chimpanzees. So what's really cool is if we take the genes that are changing in development, they're upregulating or downregulating in this chondrogenic zone of protection. So they're either they're increasing chondrogenic potential or they're downregulating osteoblast function. And we look for the regulatory elements as a class that are most changed between humans and chimpanzees. We get genes that function in the negative regulation of developmental processes. So these are genes that have a heterochronic effect on development. And all of us in this room likely know that over the course of human evolution, we have slowed down our development relevant to other primates. We have slowed down in utero development to increase, for instance, brain size and body growth. And we're seeing this process play out on the set of regulatory elements that are most changed between humans and chimps in these sets of near the genes that are affecting this anterior growth zone. So this is kind of a cool way of looking at the set of genes and regulatory switches entirely. So comparative genomics allows us to make sense of the regulatory regions that are affecting the human pelvis, and particularly identifying those that are under selection, and also those that most likely diverge the most between humans and chimpanzees for key aspects of pelvic development. We also want to know what happens in these regulatory elements in living human beings. So everybody on this call, is there a sign of this past selection or current selection going on? And why is this important? Well, you all know in this room that Darwin was super interested in understanding not just between species variation, but particularly within species variation. He was fascinated with it within species variation because the ultimate source of variation comes from everything from within a species. So are there effects of natural selection on the within species variation that we see in pelvic shape and structure in humans compared to other apes? So we bring in human genomic tools along with comparative genomics. And to do this, we acquired thousands of human genomes across the world and also ape genomes from the Great Ape Genome Diversity Project. And what we can do is for every regulatory region in the genome that we identify, we can look at diversity. We can look at ape diversity and human diversity and ask, what do the patterns look like? So we compared the regulatory element sets that are shared across the pelvis, unique to the acetabium, unique to the pubis, unique to the ischium, unique to the ilium, and looked at chimpanzee variation across 30 chimpanzees and asked, do we see differences in the behavior of these sets in each of these regulatory element regions? And what we found, no. If you take ilium regulatory regions, the diversity levels in chimpanzee do not differ in the ilium compared to the regulatory regions from the ischium and so forth. No differences. If we do the same thing for humans, we actually see that there's not a lot of differences between pelvic common or acetabulum or pubis or ischium, but we know that the ilium regulatory region, that set of regulatory regions that's unique to the ilium, that they're actually depleted in genetic variation. They're less, markedly less. And they're actually much less than what you see in chimpanzees. So this asterisk also denotes a statistical significance to the diversity levels compared to chimpanzees when correcting for population size. This tells us that the ilium in modern living population, living humans, has regulatory structure that has a depletion of genetic variation. There's not a lot of variation. So we asked morphologically, do we see the same patterns between humans and chimps? So working with a tremendous number of wonderful colleagues who study the morphology of the pelvis in fossil and living apes and humans, we actually started measuring parts of the pelvis using three-dimensional morphometrics. And so if you look at iliac breadth, this key measurement in humans, what we're finding is that iliac breadth measurements have very reduced amount of morphological variation, but they also have a reduced amount of conditional evolvability. This is the amount of variation that's left for selection to operate. In other words, it reflects past positive selection. And you can compare this kind of this amount of variation to what you see in chimpanzees across the iliac breadth. You see much more variation and more variation for selection to operate on. So this kind of matches what we see in the ilium regulatory regions, low diversity compared to chimpanzees. And if we take another pelvic dimension, like ischium length, for instance, humans actually have more variation that's remaining for selection to operate than chimpanzees. So it actually suggests that the ilium is behaving in a very unique way. And of course, we found this from the genome-wide architecture of the trait. So we also wanted to then say, how much is the ilium regulatory regions being affected by selection? And what we did was we developed on kind of a background genomic distribution of variation in the genome. So how does this work? We basically sampled thousands of human genomes and across the entire genome and made a background distribution of what variation looked like. And we annotated onto this variation known functional regions. So for instance, we probably all know that genes are made up of exons, and you know that exons are usually constrained. They're not going to have a lot of genetic variation in them because a mutation in an exon will affect that exon and that protein everywhere that protein is made in the body. So they're under kind of constraint. And in fact, in our distribution, exons are highly constrained. They fall to the far left of this distribution for genetic variation. On the other hand, repeat regions in the genome, regions that evolve pretty rapidly across from one human to the next, are highly enriched in genetic variation, and this is expected. And then we have other annotations here like transcriptional start sites that sit just to the left of the normal distribution. So what about regulatory regions that are found in the knee and the pelvis? Where do they sit in this process? Well, regulatory regions that are shared across the knee or shared across the pelvis fall right smack dab in the middle. They're not enriched or depleted in genetic variation. They're neutrally kind of evolving regions. But what about the knee? Well, the proximal tibia, regulatory regions unique to the proximal tibia, are highly depleted in genetic variation. We reported on this in Cell in 2020, as are regulatory regions unique to the distal femur, highly depleted in genetic variation. Well, what about the pelvis? Well, ilium regulatory regions are falling even more constrained than the knee. So regulatory regions unique to the ilium across the genome show less genetic variation than the knee and far less genetic variation than even transcriptional start sites. These are highly constrained sequences. In other words, Mutations that arise in them are not necessarily tolerated. If they were, they would be higher in frequency, and this line would be further to the right. Well, what about other parts of the pelvis? Well, the pubis is slightly depleted in genetic variation. The ischium is right in the middle, but the acetabulum or hip socket, the regulatory regions involved in the acetabulum or hip socket are highly enriched in genetic variation. So on one side, we have ilium regulatory regions and knee regulatory regions depleted of genetic variation, and we have acetabular sequences enriched for genetic variation. So in other words, there's evidence of purifying selection or constraint on knee and ilium specific regulatory regions, and the relaxation of this selection on hip joint or acetabulum sequences. Why is this important? Well, morphologically, we want to see if there's a correlate to this in living human populations. So working with our same colleagues, we studied different archaeological and human populations across the world using morphometrics on the pelvis, on adult pelvi. And what you can see here is that for iliac breath, there's a remarkably low amount of variation present from one population to the next around the world. In fact, this is pretty much consistent. There's not a lot of variation from one population to the next. You can also note that within a population, the error bars aren't that very large. And this is, again, a measure of conditional evolvability, how much variation is available for selection to operate on. So very little, indicating that past positive selection and current purifying selection is at play. Compare that to acetabulum height, where from one population to the next, there's lots of genetic morphological variation. There's lots of variation within a population, but from one population to the next. Now, what we see here morphologically in terms of constraint on pelvic breadth or relaxation of constraint on acetabular height, remember, we also saw here on the genetic level of the genetic regulatory regions that control the development of these structures. So we see a genome-wide, phenome-wide map. So let me summarize this point. Ilium-specific regulatory switches show evidence of recent purifying selection in humans. Variation that arises within them is not tolerated, and so we don't see variation that often. We also see the same patterns for the distal femur and the proximal tibia. On the other hand, acetabulum switches that are involved in acetabulum development have more mutations than you would expect by chance because of relaxation of selection. And we see the same patterns of variation in anatomically specific regulatory regions that we see in the patterns of morphological variation corresponding to each anatomical region. So we see genome-wide and phenome-wide maps of variation behaving in the same direction. So what are then the impacts that this past selection has had, not just on our kind of the evolution of bipedalism, which I've kind of talked about already in the today, but on potentially deleterious effects later in life in our disease risks? Is there an effect of reducing variation in these structures or allowing variation to persist in these structures? So far, I've told you how we use all of these tools here to kind of give us a context for understanding the evolution of bipedalism at the developmental, at the gene regulatory levels, and of course in the context of human evolution. What impact does this process have on our disease, on our biology today? So we can borrow from the last tool, which is medical genetics. And so one of the diseases which is really common across humans, particularly in people who are over the age of 50, is a disease called osteoarthritis. So this is the degeneration of cartilage of the knee joint or the hip joint. In fact, it is probably the leading cause of disability in the Western world. In this room, 40% of you will have knee osteoarthritis by the time you're 50 years old. And it'll cause a lot of comorbidities like heart disease and gestational, like the diabetes, and many, many other issues. So it's a really important health burden. And if we look at osteoarthritis of the hip and knee in captive or wild chimpanzees or wild gorillas, it's actually very low. And so humans have a highly derived risk of getting osteoarthritis both of the hip and knee. And in fact, risk of osteoarthritis of the hip and knee is about 40% heritable. So there's a genetic evolutionary component to it. So we asked, can we compile data from disease studies on osteoarthritis and ask, are there regions of the genome that are enriched in genetic variation that causes disease. And so there's a technique called genome-wide association studies that allows one to identify regions of the genome in humans that associate or correlate with disease risk. So GWAS data looks across the genome and says which portions of the genome are really kind of conferring an inherited risk to disease. And so using GWAS data for osteoarthritis of the hip and knee, we actually identified all the regions in the genome that confer risk, and then asked, do our regulatory elements that control ilium development or knee development, do they have more risk variance than expected by chance? And then we can ask, how does this compare to a randomized genomic background? So what you're seeing here are different tissues on the x-axis, and on the y-axis, it's whether or not they have an enrichment for disease-causing variants for osteoarthritis. So for instance, regulatory regions in the brain, they're not enriched in osteoarthritis GWAS variants. That kind of makes sense. We don't get osteoarthritis of the brain, right? We don't get cartilage degradation in the brain. So they're highly depleted. But regulatory regions involved in knee development are enriched for osteoarthritis risk variants of the knee and hip. And this was published in Cell. Well, what we found for the pelvis is that pelvic common or ilium-specific regions or pubic-specific regions or ischium regions, they're not enriched for osteoarthritis risk variants, but hip joint regulatory regions involved in forming the hip joint are enriched for osteoarthritis risk variants. And that tells us that unlike the knee, the only part of the pelvis that's kind of conferring some developmental risk for osteoarthritis is that which affects the hip joint proper, which makes a lot of sense. So recall that regulatory regions involved in acetabular development are also under relaxation of selection. There's more mutations than expected by chance, whereas regular regions involved in knee development are actually constrained for genetic variation. So with all of this in mind, we developed a model, an evolutionary model, where we take all of the knowledge across these different tools that we've used today to understand the connections between our hip and knee development, our pelvis and knee development, our hind limb development, which is critical for bipedalism, and how that confers some increased risk for disease. And this is the model. We have found, by what I've talked about today, that ancient positive selection, the HARs, those human accelerated regions, shaped morphological variation in the human ilium and the knee and hip by influencing the anatomically specific genome-wide regulatory regions that control the development of those specific regions. So we saw this by the enrichment of HARs. Now, in studying modern populations, we found that negative or purifying selection maintains low levels of regulatory diversity in these same sequences that are in the past enriched for HARs. So we have the backdrop of positive selection and the recent action of negative or purifying selection. And this selection reduces the amount of genetic variation present in the knee and the ilium, but not for the hip, which behaves the opposite. And genetic variation on both of these evolutionary backdrops, the constraint in the knee or the relaxation on the hip, confers an increased risk of getting osteoarthritis in the knee or the hip, and therefore can impact our health and our lifespan, our health span later in life. So what I've talked about today is really how we've used this integrative perspective across these tools to shed light on the evolution of the human pelvis at the level of the development and genome-wide regulation, and how that increases our risk for disease later in life. So with that, I'd like to thank all my participating lab members, my collaborators, both morphological and histological, the companies and groups that we've worked with to understand and characterize the genome-wide regulation and transcriptome of the developing pelvis and hind limb, and our funding sources. And with that, I'll stop and take any questions that you might have.
Thank you so much, Professor Capellini, for the talk. That was amazing. And yeah, we'll take questions now if anybody has.
So that was definitely a lot of material, a lot of different things that we bring together, and a nice, I think, a good confluence of data types. And I just want to emphasize that, like in all of our science, if we can develop models that are in agreement with data types, the different data types, we actually strengthen our model. And if we see a violation of that constraint, that the constraint on the model, some data type that does not agree with it, we shouldn't ignore it. We should modify our model to make it better. And so my hope by presenting this integrated perspective is that you gain an appreciation that we have to take into consideration lots of different types of data when we think about human evolution and our biology. So I'll stop there and just kind of just shut up now, I promise. Samantha.
Hi, I really loved your talk. I just was wondering, when integrating the information about collecting the morphometrics, did you have access to information like the sex, age, or if there were any behavioral or anything that might be an epigenetic factor? Because I was kind of wondering if there might be a correlation between the age of onset for the osteoarthritis between the different groups.
Yeah, so so in terms of the morphometric data, so the morphometric data was done on a bunch of samples from archaeological populations around the world. Those are independent of studies on osteoarthritis. So it's just because, as probably a morphometric person, I don't know if you're a morphometric person, you have to collect data on large number of samples to look at like integration and modularity and just, look at variation across and within the populations. So those were archaeological populations. On the GWAS data, at the level of like looking at the genetic risks, those are done generally in Western populations, in European populations, and there's been some GWAS studies done on East Asians also. So there you're sampling individuals in case and controls. Cases are individuals with the disease and controls don't, from the same population. And the background population is in this case European or Eurasian. Okay, that makes sense. And so what they try to do in these GWAS studies is also exclude individuals if they've had past injury, for instance, if they are morbidly obese, because these are constrainous factors that will affect epigenetics of a hip joint or a knee joint and the etiology of OA once you start developing OA. So they try to control for these things. Now, there are newer studies coming out where they actually don't need to actually exclude those individuals because the number of individuals is getting up into like close to millions. And that's kind of cool because then you can start factoring into your models, how does obesity affect this? How does past injury affect this risk? Are there loci that impact, risk that are dependent upon getting injured or not? So I hope that answers your question. A great question, by the way. Fantastic. Liam.
Yeah, I was wondering if you had any temporal estimates as to when the strongest selection on some of these traits may have been occurring.
That's a wonderful question. So unfortunately, the dating of genomic sequences, particularly non-coding sequences, is difficult. If we look at human accelerated regions, and we look at the regions that are falling within the regulatory regions that we found for like the ileum, right? Those regions predate Neanderthal and Denisovan. So they're at least a million years old, right? And they're still kind of fixed within humans today. So we don't know how far back they go, right? Unfortunately, that's just, you can't date them. We can look at modern human genomes and use modern ancient human genomes, which we've been doing now, and we can start putting a time frame on when variation, in the regulatory regions that are under constraint. I told you how the ileum regulatory regions don't show a lot of variation. For those sequences that do show variation, we can track what they call the haplotypes further back in time to around 30 to 40,000 years. So we have the window to scrutinize some of those sequences in the last 30 to 40,000 years. But the fixed regions like HARS, those predate 1 million years, if that makes sense. We might be able in time to date sequences by comparing like how much sequence change occurs in a given region compared to the whole background rate of evolution in the genome and develop models that way if we can calibrate it, but it's really hard. It's a wonderful question. Ideally, we would start mapping these regions that we found in the ileum and finding that they predate even like Homo erectus, like they go far back into like before even Homo, when the pelvis was particularly changed. Now, one thing I want to emphasize, and this is kind of an important point, is that we know, this group definitely knows, that the initial iliac changes that are found in like Ramidus or Afarensis are actually in some ways extreme. They're different than what we see in modern humans. So there's going to be a lot of probably turnover on the regulatory level that allows some shape to first form. And then in humans, they've done some, modern humans are different. Like we're living modern humans, we've evolved separately. And so we might have a subset of that initial regulatory variation that allowed iliac blades to go out, but not all of it. And so I don't want you to believe that all of the regulatory regions we should expect to be functional since the earliest stages of evolution of the pelvis. There's going to be a whole bunch of things happening at that point, which we just can't assess. Does that make sense, Liam?
Yeah, that's very interesting.
Yeah.
Thank you for that.
But let's see. Was there a chat? So there's a chat question which says, do you think the constrained regions could be because other factors other than locomotion, say childbirth, where there's more variable ones and more weighted towards locomotion? So in other words, so that's a very good question. In other words, could the constraint in these regulatory regions be due to, say, directly how that region anatomically affects the birth canal, right? The regulatory regions that are kind of affecting the ilium are really the upper ilium. So that should be kind of outside of that domain affecting the birth canal proper. But by intersecting regulatory element sets across the different tissues, we can actually find regulatory elements that are shared between the acetabulum and the pubis. And those regulatory regions are likely shaping chondrogenesis of the birth canal. And those regions do show constraint also. I just didn't show you here because a lot of data. And so we can see some groups of regulatory element sets that are shared across different parts of the pelvis behaving similarly or different depending upon what part of the pelvis it comes from. And we can also do this across the entire hind limb and forelimb because we have all the regulatory element sets unique to individual parts of the skeleton, and we can find unique patterns for different parts of the body. So it's a great question. Yes.
I was wondering if across the different populations you sampled, if the environments played any significant role in the shape of their pelvis or not.
So across the human populations, I can say that, iliac breadth didn't really vary that much, but acetabulum shape did. Now we know that in part biacetabular breadth changes based on population, and part of that could be due to things like body size variation. If we correct for body size, we still see that variation. So it suggests that environmentally, if you believe in something like Bergman-Allen's rule or the frame size for dietary constraint, that some environmental variation might be driving some unique changes in the acetabulum height. But ilium is behaving the same across populations for that breadth measurement, right? For conditional evaluability of breadth. So I think that, again, if we think about the three-dimensional part of the structure of the pelvis, there are going to be sub-regions of the pelvis that respond more to one factor than another. And maybe we're seeing that across the different populations that we're looking at for the acetabulum height, but not so much for the ilium. Does that make sense? We want to look at this more. Other people have argued that the pelvis has been subjected to the drift since it became stabilized. It's been drifting in different populations. And we can see signs of drift in regulatory elements for particular parts of the pelvis, but not for other parts where there's constraint. So, Dylan.
So you mentioned that archaeological populations, that upon examining archaeological populations, you could see the reduced diversity in ilium shape. I was wondering if we could take individual fossil populations like Atapuerca or Rising Star, and if we could use variation within those populations as an accurate proxy for genomic variation, or would you be confident doing that with the preservation conditions, or would it be difficult?
It's an interesting question, and it really comes down to whether or not you follow a model that says that the regulatory architecture of building a pelvis from the time we evolved it from, split from chimps, is a process that's been, it's the architectures were created and can be tweaked. And as opposed to a model where that architecture can dynamically change, right? So some traits, this is not a well worked out field. This is a hard question to ask. People who study, let's say, liver function across mammals, they might take a liver, determine all the regulatory regions for the liver or genes that are expressed in the liver, and they can see a lot of variation and a lot of regulatory element turnover. In other words, if you look at a regulatory region of the genome in one place and ask, in humans, is there a switch there? And then say, what do you see in chimpanzees? And what do you see in macaques? And what do you see in platypus? There might not be a switch there. So we can have regulatory elements that turn over quickly and genes that change in expression relatively quickly. But the question is, what time scale does that operate on? Like, is within Homo a time scale where you shouldn't expect a lot of regulatory turnover, or can you have that? Does that make sense? It's a complicated question. Can I share my screen? Because I want to show you something, for instance, that's actually pretty fascinating. Let me just get to the slide. I think I have the slide still. 1 second. Here it is. So let me share my screen. If I can get back to my, where are you? Okay. So, share my screen. Okay. So we determined the regulatory elements found in the human ileum, and we also determined them in the stage-matched mouse embryo. And if you do a multiple alignment between humans and mouse, and at every position in alignment, you say, hey, in humans, do you have a switch there? Yes or no? And then you say, hey, in the corresponding mouse position, do you have a switch there? Yes or no? Well, look, if you go across the genome, you're going to find places where humans have a switch and mouse don't. Humans and mice both have a switch at the same position, or mice have a switch. And if you compare the ileum regulatory regions, you can see on the Venn diagram on the right that the rule is actually divergence. Most sequences, ileum regulatory regions, are unique to humans or unique to mice, and there's only 2,000 of them that are shared between humans and mouse. Now, humans and mice shared a last common ancestor 50 million years ago, right? So that's 100 million years of concerted evolution. If we take the entire skeleton and look at every single site and do a pairwise comparison of every single site, the overlap percentage range is 11% to 47%. So the rule across the skeleton between humans and mice is marked divergence in regulation. You're creating new regulatory elements. Does that rule exist within genus Homo? Does that rule exist since Neanderthals and modern humans shared? Well, we don't have tissue from Neanderthals to do that. We won't have tissues from rising star. So we have to be careful how much we actually speculate. And any argument we make has to be rooted in the model that we follow. We have to be explicit about that. Hey, I believe that regulatory element turnover is the rule. I'm using this data to support that. And therefore, yes, I cannot model shape of Homo naledi population, right, based on modern parameters. Or I can if I believe there's conservation and conservation of turnover. Does that make sense? Yeah. There's a lot of work out there, and you have to be in developmental genetics. You have to be careful with it. You know, it's so loaded, like in any field, like even morphometrics. There's so much assumption that we make that goes into it that we have to make sure that our pieces agree with each other. That's what we tried to do in this study. And if you look at it, there's lots of people working on this study. There's lots of morphologists, there's geneticists, there's histologists. We're all working together to kind of come together to get some answer and to tweak our models. And our discussions are really kind of vibrant when we're together as a group, because we all come from different areas. We all don't interpret the fossil record the same way. You know, we all don't think about variation the same way. So it's but it's fun. That's the way science, that's what science should be. It's a fun interaction. Nicole.
Yeah, thank you so much for this very interesting and super rich talk. I enjoyed it very much. I'm actually quite interested in the pubis. And I'm working on this now also in a comparative context, where I find that it's very variable. I suspect in primates, it's rather conserved, whether it's constrained or it's just, you know, there's not really any particular kind of selection going on. I don't know. But I was also wondering about the anteroposterior dimension of the inlet. So from the superior part of the pubic bones to the sacrum, which using Leah Betty's data actually which I think you did too in your study with her.
She's part of it. Yeah.
Exactly. Shows that dimension is very constrained within and between human populations. And there's some speculation as to why this might be, because it's also one of the dimensions that really constrains and constricts the birth canal and contributes to the potential for dystocia, etc. And so I was wondering if you think your approach, whether all of the approaches that you talked about today or some of them could shed any light on that, given that this particular shape feature or actually dimension of the birth canal is composed of not just a single, bone. So we're dealing with pubis and actually part of the ilium also as it curves around to the back and potentially sacrum. So yeah, wondering if somehow those features could be combined and your approach still works, given that perhaps some of different signals in these bones, cancel each other out and then maybe you don't really learn much.
Yeah, so that's a great question. So we've looked into this and we're beginning to look into this more, but we've looked into it a bit. And so regulatory regions that are shared between the pubis and the acetabulum, which is actually, it's the whole structure that includes the iliac portion. So it's that rim. It's that rim. So those regions that are shared between the pubis and the rim of the acetabulum are enriched for human accelerated regions. So we see that, and they look like they're depleted in genetic variation, not to the extent of something like an ilium, but depleted. They're falling on the left of that distribution. And you saw pubis already kind of falling a little to the left of that. They were falling a little further to the left. So there is some data out there from us that suggests that pattern is occurring. And I kind of like that perspective because, again, like these regulatory regions would be kind of arguably helping to orchestrate that three-dimensional shape. And you expect that three-dimensional shape to be under some type of selection, right? The regulatory element set for all of the pelvis showed signs of enrichment for HARS. Remember, I showed you that, which suggests the three-dimensional shape of the pelvis is likely under selection. But that same regulatory element set was not depleted in variation. It fell right smack dab in the middle of human population variation. But the specific sub-element comparisons, pubis with acetabulum, like I just mentioned, that is showing a different pattern, right?
Right.
So yeah, there's things that we can do to look at that. We also would like to kind of look at, differences. We're really interested in sexual dimorphism in the pelvis.
Me too.
We really want to kind of get at that. Now, the question is, can you get at that using developmental data? And, one of the hypotheses I have is that chondrocytes, you saw that model I showed you, that was a stained human embryonic sample. And it's glaring how human-like that is at 8 weeks gestation. That pelvis is beautiful. It's actually an amazing pelvis at that age. And so that three-dimensional structure is cartilage, and it's going to be replaced by bone. Now, the question is, how do you replace a structure and maintain its shape, right? And one of the things that we're thinking about, and I don't know how familiar you are with bone development, but like there's new models out there that show that a subset of chondrocytes actually become osteoblasts. They directly become osteoblasts. We've learned in biology that like you have endochondral ossification where you get the cartilage model, and then you get an invasion of the blood vessel, and then it brings in osteoblasts. That does happen, but there's still cartilage cells that become directly osteoblasts. And our spatial transcriptomics data I saw showed that actually some of those cartilage cells are becoming osteoblasts, right? So if cartilage cells that have this shape are directly becoming osteoblasts for these irregular bones of the pelvis, there could be conference of epigenetic memory to those cells. Now, why is that important? If you are going to be forming a pelvic girdle that looks different between you, males and females because of sexual dimorphism, because of hormonal changes, those cells have to be competent to receive the hormonal signal. You can't just all of a sudden express a hormone and expect the cells to be like, hey, I'm going to turn on things I want to turn on. No. So what we're thinking is, and we want to explore this, is whether or not the early sexual dimorphism we see in gene expression is actually being conveyed over to osteoblasts that somehow primes those cells and keeps their memory so that when you get hormonal signals later in life, those cells know what to do. And we can do this in mouse model. We're trying to do these manipulations in the mouse. it's an area that I'm super fascinated with. Is there, the other thing too is we do know from late stage human gestational samples, like stillbirths, right? That there are slight male-female sexual dimorphism that already is present in utero. It's not as glaring as it is later in life, but it is there. So there must be some type of like mechanism that allows for this to happen. And we like to look at this using the mouse model, like manipulate conditions in utero to see if it translates into shape later on, and so forth.
That sounds fantastic and super interesting. If I may just make a small reply to this. So my colleagues and I had a paper out last year in which we compared, so we use a lot of geometric morphometrics, and we compared the pattern and magnitude of pelvic sexual dimorphism between humans and chimpanzees. And so what we find is that the pattern is highly, highly similar, even though the magnitude is not. And the idea kind of is that obviously there's obstetric selection on humans, but we assume much less, if at all, on chimpanzees because there's more space, and so it's supposed to be easier. So why do they have the sex differences? And then we compared that with some other previous studies that found also pelvic sexual dimorphism in similar sort of shape features in other mammals. And so we hypothesized that the genetic developmental regulation of these patterns is highly conserved, but what can be adjusted in response to selection is really how much you dial it up or down. And so we were speculating that because of course we see this sexual dimorphism in the pelvis that doesn't correspond to sex difference patterns elsewhere in the body. So how do you kind of regulate that? And I think your hypothesis of how you have these chondroblasts that then turn into osteoblasts and somehow are primed, I think this would be, it sounds like a really good candidate mechanism for kind of what we were supposing as well. So it's super fascinating.
One of the genes that we saw upregulated in the human pelvic sample, pubic sample, was a relaxin. It's developmental, and it's upregulated in utero, which is in the pubis specifically. So, like, that's just to me startling signal that perhaps I would think that when a developing system is forming, there has to be kind of like priming that's going on to test gene function, right? You want to make sure that when you're developing and when you get the right cue, you can respond to it. And if you don't get the right cue, if you get the right cue and you can't respond to it, that's it. Your game's over, right? So there must be some priming. I'm wondering if the relaxin signal that we're seeing is kind of like a priming mechanism, like turn the gene on, see if it works. Okay, it's working great. Keep it down at low levels, and when you really need it later in life, you up it. So. Yeah.
Have you published that you just mentioned about the pubic?
No, this is all kind of stuff that we're working on. My hope is that I'm going to have I have a postdoc now who's working on the pelvis from a developmental perspective. All that spatial transcriptomic stuff is great. And I want another student at one point or postdoc to work on sexual dimorphism. So, but this person's still doing that a little bit because it's in their bandwidth, Yeah. So
that's great. Thank you.
Is there a question here? The chat, I don't know, I'm missing it.
Yeah, so there's actually about 3 or 4 more questions in the chat if you'd like me to read them out, or
yeah, sure.
So I'll start with the first one. So Ian Williams asks, do you think the constrained regions, actually, did we read that one already? Sorry.
Well, yeah, that's the one I read. Yeah.
Okay, so one from Octai says, were Australopithecus offspring born facing one leg of the mother? Now, I'm not sure.
I think you're talking about rotations. I actually can't answer that question. I don't know. None of my data would suggest would even address that question. So, yeah. Sorry.
We have another from Nicole says, according to the work by Christy Luton and Mark Dabrowski about integration and modularity, the pelvis had 3 morphological and functional modules: the ilium, the ischium, and the pubis, and these modules were split from the acetabulum. After the results you presented today, people working on integration and modularity of the pelvis should also start considering the acetabulum as an additional morphological module of the pelvis.
Yeah, I mean, I would actually argue that, so when we do our work on mouse and human, because everything we've done on human, showed you, we also done on mouse. And when you look at RNA sequencing data, gene expression data, and you look across the genome at a cartilage cell from the ileum, a cartilage cell from the acetabulum, a cartilage cell from the pubis, a cartilage cell from the ischium, and you compare it to a cartilage cell in the proximal femur, for instance, and distal femur, what you see is the hip joint, which makes a lot of sense, grouping together. So gene expression patterns of the acetabulum and the proximal femur are more similar to each other than they are to ilium, ischium, or pubis, right? Because that's a developing joint. That actually, and it's been known for many, many years that babies, when that hip joint is forming, you need proper registration of the femoral head in the acetabulum. And in babies that are born with hip dysplasia, one of the treatments is to keep it locked, to keep it in traction, so you get that interaction. So from a modular perspective, it is very clear to me that the acetabulum is a module in and of itself. But modules are, again, I teach a class called Building the Human Body, where we actually look at the human fossil record, and everywhere in the fossil record, like any major transition, we address from a developmental genetics perspective. And we talk about the aspect of modularity and what modules are. Remember, modules are hierarchical structures that can be contained within each other. And so it's not surprising that the acetabulum is formed from the union of the three growth zones of the pelvis. But then it becomes a module by itself when it's involved in hip joint formation proper. So you can have modules within modules, and I think you have to begin to kind of discretionalize them based on timing, when they are actually being used in development, rather than they're always active across development. They're not, they change. They're pretty dynamic. If that makes sense.
Great,
Yes, thank you very much.
And then we have one from Christian that asks, are any of these regulatory regions located on the sex chromosomes?
Good. How you doing, Christian? It's been a while. Nice to see you again. There are some regulatory regions on the sex chromosome. And, when we saw the sex expression differences between males and females, we were like, wow, it's pretty cool. Maybe it's been driven by genes that are expressed on the X. And when we subtracted the genes that are expressed on the X, actually, we still saw them because there's an autosomal component to sex differences in gene expression that's triggered probably from the transcriptional arcade from the X itself. So there are regulatory regions unique to the X. The Y is hard to tell because the Y is diminutive, and there are also genes that are expressed uniquely on the X in some tissues versus others. Good question.
Thank you.
And the last question we have for today comes from Ian and says, in conservative regions, is it possible that the design is so narrow that there is also less tolerance for genetic variation? So there is relatively a high proportion of detrimental genes, whereas on the non-conservative, there's more variation, but the proportion of detrimental genes is lower.
So when you say the design is so narrow, I'm not sure if I understand what that means. Design is so narrow. If Ian's still here, maybe you can kind of, I think they left.
I see him right
Oh,
here.
here he is. So,
any clarification, Ian?
So, regulatory regions can target a gene. They can also target many genes, right? So it is possible that part of the constraint in sequence variation in some regulatory regions is because those regulatory regions specifically target more than one gene. If they target more than one gene, therefore they have more of a pleiotropic consequence because they're hitting whatever you do, that regulatory element can target many regulatory genes and have an effect. So there could be, but when we were doing these analyses, one of the questions I asked my student Daniel is, can you control for gene density? How many genes are near these regulatory elements? And when we control for gene density, we saw the same patterns. So it wasn't that the most constrained regulatory elements were nearest to the most genes and therefore likely pleiotropic in that regard. I hope that answers your question, Ian, on some level. All right.
Great. So we'll go ahead. Well, first of all, thank you, Professor Capellini, for taking the time today to speak to us and to answer all of our questions. We'll upload your talk on YouTube later today, so anyone that missed it can watch and learn more about the evolution of the human pelvis and how maybe about 40% of us will be heavily relying on your research in the future. We'd also like to mention that Professor or Dr. Elizabeth Sawchuk will be presenting next Tuesday, November 22nd at 5 p.m. Greenwich Mean Time to talk about the last 50,000 years of archaeological, biological, and genetic perspectives from Africa. And if you'd like to keep up to date with everything we've been doing, you can sign up to our mailing list on cambioanthsoc.com. Once again, thank you, Professor Capellini, for taking the time today to talk to us, and we hope to have you again in the future.
Thanks, and thanks for having me. It was wonderful. And please reach out if you have any questions. Okay.
Great day,
Thank you.
everyone.
Thanks.